in Europe, like chlorogenic acid. This used the medicine now still actually quite often used. Some people try to use now try to use the chlorogenic acid engineering into the letters into vegetable, leaves the vegetable, then to take then prevents the disease. And also we have other medicine like gesinocyde, the RB1, RB2, all of those medicine, and also those table list some traditional plant-derived medicine like tropine, or digitaline, and methone, morphine, quinone, actually the old anti-malaria medicine now is substituted by artemisinin because the malaria slowly developed the resistance against the quinone. Taxol now still is major clinical use, the first use. And also wind blasting, wind crystal, all those the medicine recently also developed other anti-cancer compound. I guess many of you are very familiar. So the plant, medicinal plant is very important for in terms of the metabolite, plant metabolite. This is a very old time aspirin. And also in China we have taxanum. This compound actually approved by the first single molecule compound from traditional medicine, approved by the FDA. Yeah, usually before we don't have medicine, single molecule medicine was approved by FDA just because too complex. Now the single compound molecule have very clean, very clear function, and also those wind crystal, wind blasting, and also other bottom several alkaloid or terpenoid which are under the clinical trial. So this is the sweetened, is two sweetened terpenoid compound. This fruit actually very sweet, but this is not sugar. This is terpenoid, and also licorice. Everyone knows licorice glycerin. There's some sweet taste, and actually many. This is like high-end product for those rich people who have diabetes issue, or they try to prevent diabetes. Just use this natural sweet. However, normal people will just use synthesized the sweetener. Now many of them we know have side effects, so we start. If we can large scale to produce, use metabolic engineering to enhance the plant production to produce the models metabolites, so we don't need to suffer some synthetic sweetener which have clear side effects. So all of those compounds, suggest the potential as the traditional medicine to cure the disease or prevent of disease, promote human health. Also terpenoid. The alkaloid is the first one, very strong bioactive activity, and the terpenoid is another one. Flavonoid is the most common and less effective, but because we human already adapted to those metabolites. There, I think later you are from the slides to check those compounds. And then also, besides the single molecule isolated from traditional medicine, there are also some formulary in the traditional medicine, both in China, in India or in China. Then we have traditional medicine mixed with different compounds like this one. And this, I guess this is the India, from the India review paper, from the India group. And this also, there are several. This is the Trikazu. Trikazu, I guess from the ginger, and there are several other mixed together to make into formula the medicine. All those makes the formula medicine, actually traditional medicine, folk medicine, become less popular. Just popular in the folk, but high-end or in critical the medicinal study, this is kind of a complicated, just because there are multiple bioactive components and multiple medicinal targets, and also complex magnesium. So those before is less accepted, but now we just, along from metabolite profiling, we can confirm actually there is no other component. Even before the compound mixed, and also after cooking, after the boiling in water, making into the soup to drink, the components change. Some decrease, some reduce, and maybe some generate the new compound. That's the whole process. In China, we call try to modernize the traditional Chinese medicine. They just try to start from a single herbs, do the metabolite profiling, and then after mix together in the bowl in pot to heat up into the soup, very thick soup. Then they also study what's the compound eventually getting to the human body. Then we after take, how about the gas test, how about take up in insulin, in the gut, take up those metabolite, which metabolite will take into the human body. And then we study in how many hours taking the medicine, the metabolite in plasma, what concentration change. All those use the metabolite profiling, use NMR, those non-destructive method, or use NMR usually use the study for the animal tissue. For example, we use the rat as a model to study feeding those traditional medicine formula. How about the effect? How about the metabolite? Later so we will take some example to explain how the metabolite used for the medicine from beginning to and also not only for standardized, because this plant can be produced in different area. So which area is really good? Because different environment can cause the medicinal plant components, the really bioactive components, the level different. So those make the standardizing our the quality control very important. Not all the formula contains desirable components. So that's why use those metabolites to do the quality control and tracing the metabolite change before before the mixture and before the cooking and before the souping, and then also after human take up, what's the behavior of those compounds being absorbed or being metabolized in the plasma or by the gut, by the human gums. So all of those need to use the hydrogenated the LCMS, LGCMS to study those processes. That's how the beauty actually is the metabolite profiling used in the medicine, the traditional medicine. Okay, so this is a kind of now LCMS analyze first is the mass spectra. The bottom actually is the UV detector with a different absorption, which because many of compounds like many compounds actually have absorption like 280. 280 nanometer or 350 can at least identify some flavonoid or other metabolites. So that's the goal achieved by the DAD or PAD detector. So usually after we got a lot of data, got the total ion peak, we will organize those retention time or we don't know the compound, but we first list those retention time and with the ion peak. MS, if you use pattern mass, you will get the mass 1 or mass 2, MS1, MS2. So those data will basically tell you very detailed information about the compound, the precursor ions after fragmented into the small daughter ions, which were detected in the ion detector. So those, I think the first step, we list those, and then we can check with different database. The day before yesterday, we talked about a lot of database, like a metabolite analysis, or most have vast database molecular library is NIST, NIST, is like a US government supported the database collection and also normalizing standardization. That's very important. Just like CAS, when we study chemical, you just give CAS, probably in different places have different name, but they should have a unique CAS to make sure the compound only refers to this. So this standardizing is very important. For example, this compound, this table just to show how we list our compound. Some maybe we don't know, so we just not list, just fill up all the information. Then you will step by step to fill up the gap, unknown information. Then finally you get the whole compound list, which identified in your MS, yeah, LGC or SCMS. So those lists are very important. Clearly from the ion MS doesn't matter in positive or in negative mode. In different mode, you will see those compounds. Later I will tell you, just use flavonoid that before I promised, because we study flavonoid a lot. So there are some rules, basic rules can follow up to through those iron number, we can basically judge this is the flavonoid or this is the other different compound. But however, as long as you list those ion peaks, you can find those some compound from NIST or some other metabolite database. So that's very helpful. And also, as we know, we also have a mono metabolite analyst. Those you can just throw all your raw LCMS data to there, and they will give you the preliminary prediction of those compounds. Then you can adjust or you can verify those compounds one by one. So anyway, all those steps is very necessary even for your publication. This is required so other people can as a reference to judge the compound. So metabolite. The metabolomics, I mean, just use the usage in the traditional medicine, how they for example, this paper just study one of soup, actually they call the 黄连碱毒汤. It's like several components mixed together, then the cooking in water, and finally you get the soup to drink it. Yeah, actually this used for like 1,000 years. So indeed have some effect. Now people try to, reconfirm those, use the modern technology like metabolite profiling and also use rat. But 1,000 years ago, we don't have rat, those experimental model to confirm the biological effect. This compound actually can mainly on ischemic stroke, means you have blood, you have wounding, lose too much blood, then probably, particularly for brain, is a very danger. Actually, in China, one of a single molecule compound extracted from parsley, from parsley seeds, actually was registered as one of the medicine. So, just use this as an example. So those from two different plants. One is the root, another is one is the traditional medicine, actually used a lot, and another just for benefits the brain. It's a very strong smell. To put together, it has a very strong smell. So those usually can refresh your mind, probably help. But anyway, use the rat as a model to fit it, then do the metabolite profiling, then also evaluate several items to confirm this really can help reduce the damage to the ischemic shock. They use the NMR as a technique to analyze the animal tissue, the rat tissue inside. After grading, then just put in NMR, you can analyze many amino acids or feeding, after feeding over with the control. So they get some data. Yeah, just as this morning, Dr. Winner told, like PSDA analysis, those normally the data, PCA, the principal components analysis, and also other different type of data analysis to confirm those the analysis, those data are very good, should be reliable. This is very important. That's why later we will also tell about this bioinformatics application in metabolomics analysis. And this they confirm some compound like amino acids or different metabolite, phenolic compound. Yeah, mainly, and because this is already in the animal tissue, all those compounds, after taking up into human beings through the blood to the liver, they will metabolize, modify, like in tea plant. We know tea like a catechin or theanine. All those need modified, methylated, or use glycosylated. Those modifications can change the metabolite a lot. Not only we know some metabolite actually in gut test. In our gut, actually, those microbial in gut already start to metabolize those. Some of the effect actually generated there. That's how many people believe that. That's why the gut microbiota study is very popular. But this is another L07 is another very famous Chinese prescription compared with the single component. There are several plants, including ginger and also Angelia senescens. Yeah, Angelia senescens, and also licorice, and all those compounds. I think this is the method. Yeah, they put together the formula medicine called L07. Yeah, so they also studied those use GCMS profile, because many of those compounds actually have a very strong smell. That's how the sesquiterpene or monotropine actually mainly work on our stomach, or even some work on the brain. So this the profiling. They also did the data analysis. They showed that the behavioral and biochemical analysis indicate that L07 has obvious anti-depression activity. The L means very relaxed, so that's why the medicine name called L07 is kind of just relaxed, and that's why target on the depression. But depression, very serious, particularly for the developing country. Developed country like the USA or Europe. Many women or those the symptom of disease starting show up before coming to the attention of most people today. But before, we never have such pressure to get depressed, just because the social society of human civilization development being the machinized, I think, because the depression study in recent years very become, I think, a hot area of medicine. So I will quickly show that the general rules for MS spectral feature in fragments ion peaks, mainly in flavonoid, and later on also touch other metabolites. For example, there are several for flavonoid A-glycan. The main fragment pathway is RADA. This is widely accepted we have ACD, flavonoid ACD. Yes, 3 rings, but in middle, C is the heterate. A and B is separated from malonyl CoA, and commercial CoA. Yeah, 3 commercial CoA, then rearranged, so they have a clear. So the middle C will be broken by the RDA rule. They will generate the A and B, the rings. The RAD cleavage in the C rings was denoted as the MA, MB, the different rings that generate different fragment ion. The fragment ion is different MZ values between from various RDA cleavage pathway will provide information on the number and nature of substitute in the A and B rings, hydroxy or the sugar. So the most prominent product ions, which like the flavin, deuterium, you will generate the A and B different is 155 and 133. So this for these 2 rings in respectively. And flavanol, like naringenin, generate 151 and 119. So if these put together, if you see the fragment together, basically it should be naringenin backbone. Even they have a glycosylation or others in the group. And flavanol, the camphophora, they generate 193 and 121 for A and B ring separately. So we will have figured. Their charcoal, the are different from other flavonoids. Those differences is they don't possess the heterocyclic C ring. So ACD, the C ring don't have. However, when substituted with the C primer 2 hydroxy, C primer 2 is B ring, but they will have an effect on those heterocyclic rings, C rings. The charcoal will transform to the corresponding the flavanol, their fragment in flavanol, the behavior fragmentation in A and B use the RDA rule. The furthermore, a methyl like a methyl oxygen group in A or B ring or at C7 or C4 primer. 4 primer is B ring. The C7 is the A ring. The position will readily undergo the RDA cleavage as the bond of 1 and 3 and also others. So it's boring, just that this figure, just one paper shows how much how many paper published with the LCMS Q-top. You can see how I think the LCMS tedium and also tedium mass and Q-top was widely applied compared with the other LCMS, and also this flavonoid identified the most commonly is the flavonoid, and then lipid, and then the other phenolics. The publication also solves after we have so many facilities available to identify the metabolite, because both in the food nutrition study and in traditional medicine study, both in quality control and in the medicine effects on human beings in the preparation and after taking up, and also metabolites, catabolism process as a human. So those are some information about basic information about the side chain, like glucose or galactose, if they attach to the flavonoids. So if they broken down, what's the fragments will be generated, like 162 or 160, because those are the neutron, the molecule. So you really don't see, but you only see the ion, charged ion. So if you see the 162 minor reduce or reduction in mass, the number then probably you lost one of those 6, actually, the hexanotes. All the hexanotes have similar, but the patterns or others have different. Like those one, or like retinols have a doubled, have a 308 and 487 for other, the complicated. The gallic acid have 152. Those, the typical multi, if you keep in mind, then will be easier for you to find out if I calculate the number and know, okay, this iron peak is precursor peak, and this fragmented peak just have a 162 difference. That's usually means those hexanols, either gallicose or glucose. You don't know which one, but the molecule definitely tell you this those. And this also a kind of a demonstration of, I think this is from different catechins. Yeah, catechins, backbone, camellia, synthesis, camphophora. They're so complicated. In retinols and retinols, 3' primer also have a galactose group. Then cephlavin, the 3 and 3' gallate also have those kind of fragment behavior. If you, in your identification of metabolite, you definitely have those kind of references. And this is also a kind of another type of list about the fragments and the retention time, and also the number, the list. I think all those, if you can do some during the practice, if you have those table list on your hand, so will be very easier to get the match, because those flavonoids are very common in most plants. So this is some rules about how we calculate like beauty area. Yeah, in the previous slide we see the loss of small molecule of radicals from M plus, either positive plus minus proton means positive or negative. Negative charge and used for identify the hydroxy substitute group, like hydrogen substitute group, such as 18 dark 18 lows means you have H2O lows, and 28 means carbon and CO, and yeah, others have this table demonstrates clearly how the fragmentation take place, because there are several possibilities, but each time you only have one way to generate point. But just those representative molecule like 217, 175, yeah, usually coupled together. You see, so you know this the way of cleavage actually take place, or in other places. They don't use all those way usually. You have mainly because the weak iron peak ion will be not detected, only stronger way. So this that shows there are 2 and 3 major way, or 1 major way of cleavage take place. That's the only thing. This is for flavanol, using the camphophora as an example. You can see the common CO2 or CO1 or B-ring completely broken down. They will reduce 108 B-ring. And if you reduce 28, they will see definitely the CO loss. Then generate this, from this to generate this. Then you can calculate like a 301 is precursor ion, and the reduce 28, yeah, generates the 273. And all those ways, and charcoal, yeah, just like previous, if they have a hydroxy group, they will make the middle ring, the behavior like a flavor. So they still follow the similar rules 119 and 103 for A ring and the B ring separation through the RAD. Break the possibilities. So this, I think the information very clear. This from review paper, they organize all of those. Very helpful for us to identify the molecule just through the ion data what you have generated from Q-top MS. This is a methyl flavanol or methyl flavin or whatever those, because the methylation of flavonoids is very often detected in different plants. So after you enter the methyl group, what's going to happen? And definitely, for example, here have two methyl group. So they will reduce the two methyl group in one way. You generate the 255 from 285. Then continue reduce the 128. You will generate the 227. And those ion peaks you could show up together, then you will definitely see it should be a kind of a methyl group, the flavanol or methyl flavanol or methyl flavin. Yet there are another kind of a modification is the perennial. Actually, perennial flavonoids is very, very important. Not only because they're not common, like in legume plants, they're perennial. In one of the flower, beer flower is called use flower used to brew the beer. You know that plant? Yeah, that plant actually have a lot of perennial flavonoids. They have very strong bioactivity. The fungi have very strong anti-fungi activity. So with those perennial-related flavonoids, the plants have a very strong resistance against the root, the fungi. So in legume and also other legume forage. And also in pureine, we know that in pureine also have some kind of, of course pureine is the C glucoside flavonoid. So later we will talk about the CO glucoside. C glucoside, O glucoside are very different, and some compounds have both C glucoside and O glucoside. So that's a big difference. But before, I once told you that the way to describe, to distinguish the C glucoside or O glucoside in flavonoid structure backbone is doing the acid hydrolyze. If you want to make things simple, just use the acid like 1.2 millimolar hydrogen chloride solution in 80 degree water bath for like 1 or 2 or 1.5 hours. After hydrolyze, you can log on the normal HPLC. You will detect, see if there are any change. If the compound not change, that means this should be C glucoside. Otherwise, it will be the O glucoside. Of course, this because after hydrolyze, all generates A glycol, those flavonoid backbone. So this is for C glucosyl, glycosyl substitute the flavonoid, the structure, for example, that's in 7th position. Purine, yeah, purine in 8 position, C8 glucoside. So the very the typical, they generate different ion that have a different ion peak. Yeah, so I will not just repeat those abundant precursor minus HO2 and also CHIO or minus HO or CO. Then observed in 6 C glucoside, whereas in the spectra of 8C glucoside isomer, a similar loss of water occurs within the 7 hydroxy group and only the M. The precursor ion minus CHO, loss of CHO, those are fragment ion would prevalent among the precursor products, like end products. The more fragment pathways such as loss of C2H6O3, like 7-8 dark, or CHO2. Yeah, H2O, yeah, occurs as a cyclinized 8C glycosyl following the success HO2, H2O loss. So those rules actually very widely observed. As long as you have 6 or 8 the carbon glycoside. This O-glucoside is very common. The most of those flavonoids are O-glucoside derivatives. So in case of O-glucoside, MS either positive or negative after fragment unstable, the fragment generates the neutral ion. The neutral molecule is like sugar moiety. 162 will lose. You cannot detect the sugar moiety, but you detect other ion. That's the ion. What you see is like from hepcidin, the like 611. But after remove the 1, this is the 162 is the pentose, right? Yeah. This is the dihydro. Yeah, dioxide pentose. Then pentose is just 132. Yeah, these 3 types of flavonoid, hexanoid. Another is deoxyhexane, or actually also have pentose. Just 6 carbon or 5 carbon, or some deoxide, the 5 carbon, 6 carbon can generate different neutral ion and neutral molecule loss, and they will have a typical. If you have a 162, we know this hexanoid loss. If hexose loss, if you have 162, then definitely should be the O dioxyhexa loss. Then 132 is one of pentose loss. So those is one of easier way to see the molecular loss in the ion peaks, so that you can figure out what's the precursor or what your metabolite could be. This is an example, narrative. Narrative is have a ACB 3 rings, complete rings, narrative. This molecule now have a dioglucoside. So one is dioglucoside actually have the molecule is 578 after loss after the RAD cleavage, the generates 459, and then reduce the 1 of 146, then generates 313. If you see those iron peak number 177, 107, and 271 is very common for flavanol. 271. Usually you can see if you see this iron peak, basically the typical you can see. You can judge this peak. Most likely is like narcotin, the flavanol. Yeah. So very. And also this from RAD cleavage generates 151 and 119. This is typical, the flavanol. RDA to breakdown. So those flavonoid studies much more extensively, and so many references can be very helpful. And also, if like this paper, if you review all those, we can really use it for analyze our MS spectral data. This is our publication. One of we studied in soybean the seed color. We know the several transcription factor. At least here we have MIB-A2 and also GM glycerol, glycosyl. Another is MIB-R. The soybean, two genes regulates the anthocyanin biosynthesis and determine the seed coat color. And we determine their function through the transgenic soybean hairy root. Yeah, we all express this thing. Usually, soybean hairy root don't have color. However, if we overexpress this gene in soybean, this generates color. This is purple color. Then we extract the anthocyanin through the mass spectral analysis. We will find those molecules, those ion peaks. Then we can very easily according to the ion peak and also their fragmented other ion. We basically can define those compounds, the molecule. So in detail, you can see here the 4 to 7, all those. This because it's anthocyanin. The difference in that fragment. In the previous slides, we did not show the anthocyanin, but later on we will. But you just use this. You can see so the 475 is most likely mixed the peaks because both have 475 and also 452 fragment and 476 fragment. So we saw probably the pelagology 3 glucoside plus also have an acetyl glucoside. Or another is cytidine, because those molecules are the same or mixed. You can actually see they're mixed. If otherwise, you will see that definitely this should be the like A6. We can see this is the pneuridine 3' dialglucoside. Yeah, we also use the hydrolyzed, use acid hydrolyzation, hydrolyze the prepared sample. Then we can see that use the normal HPLC. We can check with the A-glycan because A-glycan is relatively simple, like pelargonidine, delphinidine, cynin. So very easy to check the basic backbone. Then we can use the mass spectrum to define their side chain, the modification. So this is the ESA mass fingerprint of purocyanine. There's some purocyanine in larger molecule after the broken down. They have the other different ways because if the metabolite very large, but still finally they were broken down. So they still follow the RDA as long as they have a flavonoid the backbone. So again, we just use one of a single mass spectral the data. This is good. The total mass spectral data for iron peak 579. How do we see judge the fire? This iron peak, like here, labeled with from 702 to 576. 576 have 162 neutral the molecule. Neutral molecule loss. Then also have 288. Then probably have other components together generate the different peaks. So we can actually predict what's for example minus 152 or minus 152. We know this is one of RDA generated the fragments in B ring. So those we can predict. That's RDA fragmentation rescue the consecutively with the M152 ion respectively. Then, but another is 451 was derived from the loss of ring A and the sugar unit due to the HRF fragment. Here shows the MRF. If the lost class of 1 sugar moiety then totally is 288. Yeah, keep in mind, those are actually very good reference for your analyzing our from the database checking, help you understand the metabolite. This is another example. Also show the similar way, like there also have a MS2 fragment or MS1. All those generate from helium MS. The catechin, the dimer, is catechin dimer, based on the precursor ion at the mass charge ratio is 577, as well as after MS2 fragmentation, which results in the product ion at 4 to 5, 407 and 272 over 298, as well as 228. Those fragment ion, they're consistent with this supposed literature. That's why we always challenge you need to have literature, take a reference, help us to digest those. Unless you use the NST, those compounds is similar to the the cation dimer, to the MS the data. Actually, you can check the reference to show this actually is a catechin and the epicatechin together, the dimers formed. This is another, I just saw the paper have so those kind of data. You can see the retention time. The retention time is vary in different machine and also different mobile phase in LCMS or in HPLC. However, the molecular ion generated always the same, the phase according to the first generates the 287 and then that generates the 245, the catechin. For epicatechin, it also generates because the same molecule, just different. And the 577, just last slide, so we show that generates 407 and 289. 289 is the catechin. Yeah, 289 is the catechin. The 407 just the medium. In medium, there are also the non-complete fragment. So all those have a 289 fragments. That means all of them have a catechin or epicatechin, the signature ions. But for the larger one, the 289 fragment is very small. They're just like the previous, just like previous slide. In those larger one, you cannot see the very unless they completely broken down to generate 289. Otherwise, they still in the complex like a glucoside. This is a glucoside, right? Have some still. So the larger molecule is very difficult to continue broken down. That's why the OV trap was used. This high field generate a complete molecule to make sure those fragments are all present, so we confirm 100% confirm this molecule is what we expected. This is another one actually for the epicatechin or EGCT. This major catechin molecules take place in the key links. And this is also the catechin hexyl. I think this mass spectra also good, shows the real, the 451 is the catechin hexyl. So they generated 281 and also generated 245. This one, this two ion peaks is typical. The characteristic products ions can be used for those catechin or epicatechin hexons because those are same, basically just the formula is the same, but configuration is different. This is for like a simple phenolic compound that have different ion the peak. So I I think you can use it as a reference, chlorogenic acid or other commonly found compound. The succinic acid, this in LCMS can analyze many small ion peak. Can be a reference. This also MS for total TIC analysis. Usually, I think this is after you put on the ion selection. I want to search for example, I want to search the 455 ion peak. Then they will give out those relevant. Those 455 they come from where? They will show up at the same time. You will pick up ion. They will show up at the same time. So you know this fragment actually is a fragment ion from other larger the precursor ions. Yeah, this is another compound. Actually, they also this is the total ion TIC, and below they did the detailed analysis, and finally in publication they show up like this. The total ion peak to show the and in a similar way, some publication would like to show the metabolite like this. This is try to analyze one of natural products. Use the LC top to clarify some flavonoid. I think this is from YCHP, the kind of a medicinal plant. This is for saponin, for ginseng. Yeah, for ginseng study. Yeah, the way of this the wind. Yeah, also one of very common wind diagram analysis tools, also very important. It's just compare with the different effects in different organs. For example, in liver, take the ginseng, RGS, RG1, or most significant compound is RG1, because there are so many different the saponin. This is the different variety, and to show that how according to literature or report, okay, RG1, RG3 have a different preferred the medicinal function mechanism. Of some probably prefer to improve the memory, or some improve the liver. But anyway, they did those kind of literature analysis, and also according to the ginseng cultivated in different areas, or even through the Korea, North Korea, they also produce those ginseng. American also produce those. How about those? Put together metabolite comparison, and each metabolite have a preference, the working mechanism, then we generate those very comprehensive data. This, I think, just the non-target metabolite profiling, and use so those kind of the metabolite comparison, you not only use for standardization or for quality control. If you have one, for sure, government already clearly see that the medicinal plant growing in this area can be as a control, the good. Then you can use this metabolite to do the similar profile, mass spectral compared with the others. This is like total ion, or just according to different retention time, like HPLC or UPLC profile, not a total ion profile. So you can make a comparison, preliminary comparison, even can tell you the much more important data, roughly. Like yesterday I told you, if we do the QTL, actually we really don't need, at the beginning, don't need to spend so much time to identify each compound. Now, first we just compare all the, like those non-target metabolite profiling, HPLC or UPLC, the initial. profiling the identified compound. For example, in those, you need around detection time of around the 14 to 16 minutes. There are two strong peaks show up in RS or ZGS. Yeah, those definitely different from others. So if you find some, yeah, usually it should be very similar, but those different anyway show very dramatic difference. Similar seems not same Jason product, but I think those are very. Now, the many scientists, one side try to study digging into the particular metabolite, but another side is try to roughly generate a large scale, use from a whole view to see the metabolite change. So that's also gives, if you have larger number of biological samples, those will be helping you focus on some of them to show the difference, then to dig into the more details. This is also the ginseng section, because this is for medicinal plants studied a lot in China. People just believe those metabolites have the miracle effect. This is in different organ. This is actually in flower and also in different stage of you can see the metabolite profiling. Also, I prefer those receptor time difference. You can just from first looking, you can tell the difference. Yeah, those difference you compare different organs. You basically have much accurate information before the mass spectrum. This is like a LCMS isolation. This is kind of a bioinformatics analysis of those metabolites. See if they can class together, the data reproducible. Yeah, you normally in different organs you should class them together. If you have 4 or 5 repeats, you should class together. That means your profiling is very good. You no machine problem, no your sampling problem. You did not mix the sample. Yeah, this you use those PCA analysis basically tell you this. That's why for chemical analysis you publish in food chemistry, you publish in food research international, those journals they ask me to do the PCA analysis or other the OLSDA analysis, so completed a different set of analysis to make sure your data generation is great. Your different treatment or different group or sample is reproducible. Yeah, this also uses a different column to process those metabolites. You can see the isolation is very different. Different column, RT column, carbon of different brand. Let's show the difference. So what's the time? How we still have?
Yeah, it's 11:30. So you can continue for 5 to 10 minutes.
Okay. Yeah, absolutely. So
otherwise you can take the coffee.
okay. Yeah. So this I'm trying to quickly go through the previous. Yesterday I have very important part I did not finish because I think this part very important. Particularly for young generation should be ambitious for the to embrace the new biotechnology to the innovative research. Using the useful metabolite as a target to fortify the nutritional value of medicinal function of plants. And using the key metabolic gene or transferring factor, various genes to achieve the metabolic engineering goals or genetic improvement. That's also for the breeding. If you have those kinds of magnesium unwieldy, so those breeder, they can find the marker for corresponding the gene, then to use this marker to select. Very quickly select, just like this professor generate all of mutant. We want to. I think many years ago, I heard one report in England actually one group focused on the artemisinin, the mutant selection. Do the mutation, seeds mutation, then the girl saw those seeds on the field, then generate different plant, then use the mass spectral scanning, or you just use a very simple, quick, accuracy to measure the artemisinin level. Then find the,
compared with the standard?
To find the very high artemisinin yield, the variety. Then use this variety for the production, because today still artemisinin is one of the major anti-malaria medicine, right? Still used a lot. So the different way, anyway, use a different way to pursue those goals. So I will just use several examples. The engineering, the melonial, the pathway for production terpenoid. Actually, that the compound is Taxol, the very famous anti-cancer. Now this Taxol actually can use for many. The first one is previously used for women, urinary cancer, finally used for small cell cancer and lung cancer, and now even used for other cancer. Anyway, used multiple enzymes. Then recently, the Taxol genome was resequenced. Just this year or earlier this year, the Taxol genome was sequenced by a Chinese group. Then they dig all the enzyme and isolate the genes, express the gene in bacteria, then direct the gene to the enzyme activity as they demonstrate all those pathways. But this the previous work actually very good, also giving some the first important trial on this. This is the enhancement tomato fruit with health promoting assigning by expression of selective transferrin factor. This is John Innes Center. His marking, his marking published. That's very important. Many the world major, the mainstream report this use generally purple the tomato, and they said if you have cancer cells, you feed this purple tomato powder, give the rats the cancer rat, then you will let the rats survive for more than 2 weeks, more longer than the regular cancer rat. That's very exciting to tell people that it's this purple tomatoes then can help you promote health and live longer. For terpenoid, for the produce, there's anti-herbivore plant. So this is vitamin A. I guess this is very famous, right? Like 2000. No. 2000 is already generated the golden rice. Very reported, but now still we couldn't in the field to create the commercial over just because transgenic. I don't know the government why they have. Actually, this is free. You can use any. There are no need to license those. Golden rice actually. This is for the trichome, for trichome production, the volatile compound in the medicine or in peppermint to produce those valuable compounds. Actually use a lot as big factors. This is for vitamin C, actually also is one of the major. The vitamin A is in golden rice or the carotenoid provitamin A in other the cauliflower or other potato. Those work. This is for vitamin C. The earlier metabolic identity, the vitamin C is derived from glycolysis. And this is a recent paper published biofortified tomatoes provide a new routine for vitamin D sufficiency, because vitamin D is very important for normally we take to help our bone or help muscle, also help other immune system enhancement. The vitamin D is very important, and they did very excellent work to demonstrate the pathway in plant and generate transgenic plant, mutation plant. Then they can enhance the vitamin D accumulation in the plant. Actually, this is mainly from cholesterol biosynthesis pathway. This is vitamin E. Metabolic engineering is very earlier, but now actually this vitamin E transgenic soybean has been produced. Actually, those transgenic soybean mainly used for feed the salmon in Canada and also in American, in North American the area they have artificial the salmon the cultivation. So to generate the better color, they use the vitamin E or carotenoid, those rich carotenoid. So the soybean is like a brown, the brown color, very high level of carotenoid, provitamin, also vitamin E. Then feed those salmon to produce the high quality of the red meat, the salmon. This is the last slide. The people still trying to figure out those elements like tomato or potato or those plants generate some stereo alkali. The stereo alkali is toxic. We know the potato if they start the body. If the tomato store for a long time or in improper condition, if it start burning, they will generate the stereo alkali. The stereo alkali is kind of very toxic, so people try to reduce those in green tomato and also in potato to enhance. So we use the CRISPR-Cas9 to prohibit several pathways. There's many publication about the stereo alkali biosynthesis. Yeah, you try to get it. And then we will briefly touch some biomechanics about for metabolomics. Quickly, you Hillenberg or Hill-Carty cluster adjustment.
Thank you for the first lecture. So it will be now the bioinformatics. What the bioinformatics tool we are using for the analysis of the metabolites. So this is the last tutorial of 2 hours. In between Ritu is coming. We would like to take the exam from all of you, 10 minutes. And during 10 minutes, Professor Jim can also take the rest. And yes, we have a very tight schedule at 8, 1, 12 to 1 and then 1 to 2 p.m. So it is humble request. We would like to finish within the 45 minutes because after that, you know very well, ma'am is need to board the flight from the Matinda airport, so they need to go at the earliest from here actually. So this is my humble request. There is a few guests also come up here from the university administration, senior most professor of our university, and I also requested the Honorable Vice Chancellor let me know he is available today or not. Yesterday he was not available here, he was in Delhi in ministry for some presentation. So we will give you the certificate before that, but you have to come in a queue and you take your individual pics because these individual pics we will share on the Google after some time, Google Drive. But we also get this one in this document. You take 10 minutes. So, yeah, give me this mic. I would like to acknowledge that we are giving you one participation certificate, one grade sheet, as well as this photograph of the inauguration session. So yes, there are 4 to 5. Previously we fixed the 50 numbers, but last day we got the 5 more entries for the participation from our university. So the participation certificate was the 50, but this grade certificate is the 55. So we selected to whom right now we are not giving the participation certificate. They will get tomorrow because they need the prints from the another place. Otherwise, this document is the complete. It has the 1 credit. So this Sakshi is circulating the attendance. 2 different type of the attendance. One is for 5 days in a single seat, and second is the individual lecture, individual attendance. So here please write down your email ID and your phone number very clear, because we have to upload all these things on the Gyan. And whatever the course material we have given you, now you already done the feedback. Even after I am telling you, I will sort down according to lecture the different courses. I will find out all the relevant papers and I will share within 3 to 4 days after completion of this one. Because we want to help all those needy persons who want to pursue their career, faculty to submit some project. And I am confident if you submit a small project on the metabolomics, you will definitely get it. So this is the best time to push yourself in the metabolomics area of research. And all those students who are doing the PhD, at least they think how can they utilize these experiments in their doctoral thesis. And all those MSc and just enter into the PhD, they at least assigned one objective on the metabolomics and try to correlate the metabolic alternation with their particular issue that they want to propose in their hypothesis. So this is something that is very useful in many sense. And yes, I would like to give you the time with Professor Jiang also. Professor Jiang also speak about you. And whoever I will share the email ID and the contact number of the Professor Jiang with all of you on the WhatsApp. You can contact him anytime. There is nothing like that he will not respond. Maybe he will little bit delay. I am delaying from the last 5 days. I am not responding anyone. So same thing happen when you are busy a particular task and you forget all others. So this is a normal phenomenon when you are highly focused and working on some area. And regarding this particular area, trust me, to do the metabolomics is easy, but to do the accurate, precise metabolomics require extensive times. In nature paper, a science paper requires 5 to 6 years of the robust data. So try to understand, and a single peak if you identified in the metabolomics, that's enough to recognize you all over the world if that is associated with some biomarkers. No other techniques can give you so much recognition within a shortest period of the time. And here you don't need too much machines, you just know the information about the different techniques. First of all, the different machines, how they are working, what type of the data they are releasing, they are producing, then how you interpret that data, and what you want to solve among from those data. So there is so many things we mentioned, we try to cover all of them here, and yet because this time is a little bit first time we are doing this Gyan course, and we know there are many gaps here, I try to fulfill those gaps also. It's just time filling, he's taking the rest. So now please Professor Jing come and start the second lecture. It is the tutorial. The first lecture was very elaborated, and try to understand whatever the chromatogram sir was showing here, so many peaks representing the different compound. It is not a small task. It's years of the affairs by which you identify this peak is this metabolites. And if you know this robust mechanism of understanding these different interpretations, then only you can do the real science. So that's why, yeah, please sir, now you start your second portion. Bioinformatics associated with the metabolomics.
It's a great honor to be here interacting with so many professors and with your excellent student. I think you willing and attend this meeting and the registration finished to come here. That means you really want to learn something. I hope our game meeting, our game course this time really is meaningful, and I hope you also get the fruitful the impression, our knowledge, our kind of. Also, I think you probably most of you first time see a Chinese looking like a scientist here. So those days interaction is really kind of fresh. Everything new, and for those the courses, the PPT, all this information that we get with the Dr. Winner bring to you. Yeah, I hope so. I hope this is really a very good opportunity. You open a window to see through window, just to see another side of the world. Actually, it's a very truly exciting area in future. I'm sure it will benefit both our the plant foods and particularly for India. Many people, many Indians like the plant food, but rather than the animal food. That's kind of different from China. But in China, still agriculture is very, very important part. That's why I think Dr. Winner holding this courses will apply for this and get granted by the Ministry of Education of India. So this, I will continue just express my section. This is the Venn diagram also shows a kind of technique to show the cluster, the concept is larger or smaller, what's the overlap from this group to another group. So for this analysis will be useful for distinguished comparison. And this is also some HCA. This is the first PPT. We show the hierarchical clustering analysis, and also PCA analysis. Those are very important to show your data reproducibility, and also the group by group have a very uniformly different. For this group, you have 3 repeater, or you have 5 repeater for metabolite profiling, and all those data by the principal analysis you group differently. That means the data is reliable, reproducible. Yeah. And also then used for network analysis. After you identify those metabolites, you can put the metabolite through the software like a KEGG. The KEGG includes the enzyme and the metabolite, and in a network, basically applied not only bacteria, plants, also animals. The sum of this benefit, every enzyme have a standard enzyme annual lecture, the methodology, like EC, what's 2.3 or 1. So very standard, they can uniformly apply to animal. This depends animal or bacteria, or this really depends on your nutrition. Then those different data analysis, statistic analysis, grouping analysis, classing analysis. Yeah, those PPT, I think eventually after we connect, we can send you to you. You don't need to take those. I think many of those, like the previous picture, we show the flavonoids that have different type of flavonoid, as cyanine have different fragmentation ways. So those data very important. I think you can really keep in your hand to help you analyze, to understand the flavonoids, understand the MS, the principle. Really.
Sir, last slide.
This is the last slide?
No.
Please go one slide.
Previous slide. Go please one.
Oh, this one just to see the many statistic and analysis,
Now.
the method, yeah. Yeah, this just you use the PC or PLSD. It's a different name, but basically the analysis should be very similar. PC, for example, A, B, and C. Can you see the difference? I don't see the difference. I mean, sometimes we just regularly confirm our data, reproducible, clustering, or very good. That's enough. Then we need to going on. We don't need. I reviewed some scientific papers just to try to publish those large-scale metabolite or transcriptomic or some data. They also can use it for the PC analysis. For gene expression, also can use it. Widely applied to the molecular biology or for the metabolite. But interpreting, overdoing, this is probably not good. Too many figures, too many things. You need to tell the principle. What's the major finding, scientific meaning are. If you confirm it's good, that's good. Then continue to next step. So there are also other metabolomics library or some database needs or other those metabol analyst 3.5, 3.0, 5.0, 6.0 already come out, and also other. I think all those you can use the GCMS analysis, and also those for PC analysis. The online free website. So many different companies developed different method program. You can just put your data there, generate, or just download their software to do that. The R package, those software, and also Mac data for the Mac 7 for the gene cluster or for the protein or DNA sequence alignment, all those, and also below is for HC, different type of hierarchical clustering analysis. Those analyses are actually very helpful for you quickly identify different biological sample. Some of them actually can put together both with the genetic information or from metabolite information. You can know those group actually is very similar. Biochemical or metabolite, those similarity actually indicate their genetic correlation, very close in genetic. Probably they are segregated in other place or is there some crossing. So, very meaningful. That's why the many different tools for hierarchical clustering analysis show up. And this is for metabolite pathway, the KGG, Mandarin EMT project, or other the data analysis. If you log in, put in each one of them, you will see a lot of the information. Each company, or sometimes in those magazines like nucleic acid analysis or metabolites or metabolomics journal, the different, they usually generate larger. Some paper reports that we generate large database, and we incorporate all the legume plant. We incorporate all the rice. Rice is much more information just in single plant species. We generate so many data because many people study those, and also other. So you can find the corresponding the website or databases, especially for your plant or some medicinal plant. Actually, in pharmacognosy in USA, they also have generate many different transcriptomic in I think more than 100 different medicinal plants have this kind of database. You can download to get the information for different medicinal plants. They already generate, but we just don't know. You can actually to find those database to do the analysis, even isolate enzyme. Even we don't have we did not generate our own transcriptome, but use their transcriptome the same plant, you can actually design primer to take out, extract your DNA to clone gene. Yeah, that's exactly you can do it. I think that's all of my the last. Yeah, hopefully you enjoyed and learned a lot from courses. That's what the GIAA want to reach this program.
So, thank you, Professor Jian. Have a seat. And yes, it quickly finished his part because this bioinformatics needs the skill of the bioinformatician. So, in which you have to compile the raw data. You have to collect each and every files, convert them, and then visualize them by using some particular tool which reflect the change you want to identify as a feature signal in your data. So that's why it itself is just like a whole course multivariant analysis. And if you just strengthen yourself in the multivariant analysis, so you will find out that this is some area you can use it at many places. That is not only associated with the metabolomics, you can go ahead with the genomics, you can go ahead with the transcriptomics, and many other omics also require this needs of the MVA, multivariant analysis. So this is my humble request, please you have to read about it. There are so many books. I will share one book on the WhatsApp with you. That is a Fowler book, and that is the best book I just saw in somewhere in a book fair. I just requested my library, they purchased one book on the same day, and from that day I got the idea of the metabolomics from that book only. So that book is something that was published by some Chinese author. I forget Jin Jun, that is the name of that one. And the first 4 to 5 book chapter of he is the author of that book. So those can give you elaborate information about the different techniques also. And meanwhile, this course is almost going to sum up here today at the 1 p.m. But we are here, you come here and give your feedback. You forget what you wrote there. We just compile them and send to the ministry. But yes, many programs has many issues, some positive, some negative, and sometimes we are not match up ourselves with the expectation of the you also. So this is the point that also give us a suggestion how can we improve ourselves with the time. With respect to this course, you can give your feedbacks. So I would like to request randomly initially, and then anyone who want to come here and give the feedback on this course. And right now remember, only you and we both are here. There is no another university administrator. And we are the live, and we are not afraid if you say anything, that is it. We never want to take anything on the personal ego actually. We want to improve ourselves, and we want to see the real transparent feedback from the viewer sides. And it will be good for us because in the near future, if we would like to, this was the first program, trust me, I organized in this university in the last 10 years. This was the first program only. Before that, we conducted one day interaction with the farmer by the FRI scientists. So we just arranged the lunch for them, and 4 to 5 scientists from the FRI come here. So that was with the local peoples. But this one was with the faculty and my PhD students as well as the MSc students. So I know we need to change ourselves, we need to look some more good things need to incorporate in such type of the courses. And now I get a lead here, so in near future you will find out a few more faculty from the China as sir suggest us. So we will invite them also here in the next level of this course. This was the plant metabolomics and the technology. So we can split it into the NMR and the plant metabolomics. So when we go with only one technique, then you must have to explain the raw data, you have to must to explain each and every figure how they are the constructed. It was course and it was decided by me that we have to split this course into the two parts. First, we have to explain the basics to the student. Second, we have to show the students the final outcomes of the paper. So we both work very fantastically as I observe. So I try to cover all the objectives of the basics. Even I also explained some algorithms, and they are something giving you the basic knowledge about the different pathways. And this Professor Jiang come up with the final outcomes, how those things give the data, and you are getting some figures in the tables, and those figures in the table giving the final outcomes in the form of the answer of some hypothesis also. So we tried, and it was very difficult for him. He was layover in the Hong Kong was 8 hours for him, but he come just one day before, and next day he started. First time he come in the India, so first day I think it was you were not able to understand his way of presenting, but now he improved a lot, and you are able to understand what sir is telling you. So this thing only happen when you counter with any foreigner faculty for 5 to 6 days. After 5 to 6 days with staying with them and just make the answer, you can then now whenever you meet any foreigner faculty, so you will be the comfort, at least one whoever is come from the China. So this we try to build up this confidence, so that's why we stayed him in the guest house. So all those who would like to interest, they interacted me as I thought actually, because it's not me who can push you to interact with him. All those who have the driving force, they automatically come and interact with him. He never say anything no. So otherwise we don't have so much facility, and someone compelled me to please give the stay to him to the comfort inn and somewhere 3-star hotel of the Batinda. So that was not good for the live-to-live interaction to see the things here. So that was a something essence. So now I would like to declare a 10 minutes break, and after 10 minutes breaks, 10 minutes paper, and then you have to come and give your feedbacks. We definitely try to incorporate whatever you will tell me. And frankly, we will never take anything on the ego, because we are the teacher and we believe in the positive sides of the student always. So trust us, nothing is wrong. Whatever you come and say, we will definitely try to consider for the next time. So it's a 10 minutes break, but yes, कितनी देर में आ रहे हैं वो? कहाँ गया वो लड़का?